Hello, and welcome to this GOG video podcast discussing low-grade serous ovarian cancer. My name is Dr. Kathleen Moore. I am the Associate Director of GOG Partners and a GYN oncologist at the Stevenson Cancer Center in Oklahoma City. And I'm joined by two of my very good friends and experts in the field of drug development in general, but low-grade serous ovarian cancer in particular, both having led clinical trials in this space. And so we're really excited to have both of you here. Dr. Banerjee, would you please introduce yourself?
Thank you, Dr. Moore. I'm Dr. Sana Banerjee. I'm a consultant medical oncologist at the Royal Marsden NHS Foundation Trust in London and a team leader at the Institute of Cancer Research. And I'm the global lead of ENGOT-ov60, GOG-3052, ramp 201.
Yes, which is what we're going to be talking a lot about today, which is our study in low-grade serous ovarian cancer. Dr. Monk, you have quite a bit of experience in this realm. Would you please introduce yourself?
Yeah, thank you for having me today. Brad Monk, Phoenix, Arizona, where I'm a professor of the two medical schools here, Creighton and University of Arizona. Thank you for having me. The thing I like about this shows the international collaboration between ENGOT and the GOG, where I'm the co-director of partners. So this is a great collaboration, a great opportunity moving forward. It's good to be here.
Yeah, it's great to be here. And I'm really excited. I think we all are about the increased focus on a lot of gynecologic cancers. Let's just be honest. We just all got back from ASCO, and that was an exciting time. Lots of limelight for gynecologic cancers, which is long overdue. But particularly for low-grade ovarian cancer, which we've all sort of known about for a long time. We know it's a special sort of subset of ovarian cancer. There have been some attempts at trials, but I really feel like, and I'm going to play on words, we're ramping up interest in this very important cancer that a lot of us treat. So Dr. Monk, I want to start with you because you've led one of the big pivotal trials which attempted to change standard of care and really kind of did for low-grade serous ovarian cancer. Can you just start off by a general discussion of what low-grade serous cancer is? I think we all can say it's not just a friendlier version of high-grade serous ovarian cancer. Like, what is it? What makes it unique, and why is it a high unmet need?
Yeah, thank you for that. So low-grade serous ovarian cancer is distinct from the more common high-grade serous cancers, not only in its appearance. And remember, almost basically all gynecologic cancers are rare and expert pathology review is important, but it doesn't have the nuclear atypia and is more papillary, if you will, is more calcified. But most importantly, it is not a cancer of genomic instability. It's a cancer where the RAS, RAF, MEK, ERK pathway is mutated. You're going to hear a lot about that. I want to point out one other thing. There's two ways to get to metastatic or recurrent low-grade serous cancer. And it doesn't really matter how you get there. The first way is you have a borderline, generally metastatic, that evolves into that low-grade serous cancer. Or you have an advanced primary ovarian cancer, you think it's a high-grade serous, and then you learn, oh, this is a low-grade tumor. So there's two ways to get there. But once you're there, the treatment, the systemic treatment is the same. And importantly, the systemic treatment is less effective in the traditional way. So for example, PARP inhibitors don't work because it's not a genomically unstable cancer. And chemotherapy is also less effective. And then we have some anti-VEGF and hormones. But more importantly, we can interact with this pathway, a MEK inhibitor. So 4 classes of agents, inherently chemotherapy resistant, is rare. It requires expert pathology diagnosis and also has some unique molecular signatures.
Thank you for that. So I think that just to sort of recap for some of our listeners who may be less familiar with low grade, this really is a unique histopathologic subtype of ovarian cancer. And I think, Dr. Monk, you made this point a couple times. It really does require expert pathologists to look at it. And, I think we're going to talk about, because I want to come back to one of the things you were saying, because it does challenge us to really identify these patients and make appropriate selections of treatments. And sometimes, and we'll talk about this later, when we're doing neoadjuvant chemotherapy, for example, and we have a little nubbin of a biopsy that's been taken, there's some errors there. Would you agree with that? And that's something that we have to work on.
Exactly. Mm-hmm.
Yeah. So you mentioned a few of the options for treatment. We start with standard of care, even though we know it does not great, paclitaxel carboplatin. There are clinical trials in the front line that are trying to improve that. And then we move into this sort of second line era of standard of care chemo, endocrine therapies, and MEK inhibitors. And so what's exciting right now to you for clinical trials for this patient population?
Yeah, so a couple of points. Number one, all MEK inhibitors are not the same, okay? We think that, all PARP inhibitors are the same or all anti-VEGF agents. No, they're not. So what's exciting is this new MEK inhibitor that I'm sure many of you have heard about, avumetinib. And this is a very unique opportunity because this is different. Okay, this is a unique RAF-MEK mechanism of action. It has novel intermittent dosing, and obviously it's oral, so it's convenient. It has breakthrough therapy designation in recurrent low-grade serous cancer, and it's also been added to a FAK inhibitor, defactinib. So I'm excited to hear from Susanna to hear about this novel trial called RAF and MEK program, adding these two agents together in recurrent low-grade serous ovarian cancer.
Great. Dr. Banerjee, why don't you take us through the design of what we call the RAMP trial? Brad just gave us the definition of RAMP, but it also just, as you and I were joking, it's sort of ramping up interest in low grade. What's the design of your study? Because it's not just avumetinib, is it?
No, thank you, Dr. Moore. So first of all, the combination of avetametinib, which was previously known as VS6766, with defactinib in RAMP201 is based on results from an early phase study in the UK called FRAME, which I presented at ESMO 2021. And based on the activity we saw there, which showed a response rate of around 45%, that's what led, as Brad pointed out, to the FDA breakthrough therapy designation and provides the rationale to go forward with RAMP201. So RAMP201 is a phase 2. It's an adaptive, multi-center, multi-country, a real example of the collaboration between ENGOT and GOG. And patients were randomized to either avetametinib alone or with defactinib. And we had cohorts with KRAS mutated tumors and also KRAS wild type, recurrent low-grade serous ovarian cancer. And the goal of our study was to identify the optimal regimen and the patient population to go forward with. And this is based on response rate by blinded central review. And that's the initial part A, that we presented at ASCO 2023.
Thank you for that. And so just before we move on to a little bit about what you presented at ASCO, put into context the findings of the FRAME study. I know it was an early phase study. We have to always acknowledge. We've done other early phase studies in low-grade serous. What's a 44% response rate in low grade? Is that what we expect? Put that into context for our listeners.
Thank you. I think it's really important because, as Brad pointed out earlier, the issue is the response rate to our standard of care therapies. So 0 to 14% in the NRG-281 study in standard of care arm of chemotherapy, so paclitaxel, for example, and hormonal therapy such as letrozole. Tamoxifen 0%, topotecan 0%. So that's what we have to offer our patients. Now with MEK inhibitors, which have been a step forward, with trametinib and also binimetinib, the response rate is around 25-26%. So in an early phase study, seeing a response rate over 40% in all comers, irrespective of KRAS mutation status, was very encouraging. Not only that, the progression-free survival in the early phase at the time of the data cuts of FRAME was around 23 months. And what I found very notable and very interesting was response to avexitide and defactinib in patients that have been treated with prior MEK inhibitors. And that's why I was so excited to go forward with this in RAMP 201.
Exactly. So so we presented data at ASCO, or you did. Who was enrolled? Can you just take us through the study, and we can kind of start with the demographics maybe of who was enrolled in this trial thus far?
So this was exclusively low-grade serous ovarian cancer. It was recurrent disease. Patients needed to have prior platinum therapy. So all these patients had prior platinum therapy, and this is in line with the standard of care algorithms to date. So patients needed to have a known KRAS status. So if that wasn't available, then they could be tested in order to have the KRAS mutation status. And they needed to have measurable disease per RECIST 1.1 criteria. They could have unlimited additional prior lines, and importantly, they could have had a prior MEK inhibitor. So the patients that we presented, the efficacy results from around 64 patients from Part A and over 150 patients for the safety. And that's from the data cutoff of April 2023. So the median number of prior lines of systemic therapy was 4 on the combination arm, 3 on the monotherapy, and it really represents a heavily pretreated group. 64% in the combination had prior bevacizumab, and 90% prior hormonal therapy. And patients were included that had prior MEK inhibitors as well.
Yeah, so this was really, as you said, this is a real group. This is who we see, in the community, in our clinics with recurrent low-grade serous ovarian cancer. This is not sort of a highly selected group that came on this clinical trial. And I think it's important, you mentioned they could have had prior MEK inhibitor, and they could come on with known KRAS, but either wild type or mutated to really sort it out. So that was the study population, as you said. What'd you find?
So firstly, thinking about tumor shrinkage, because for an individual patient with the burden of disease that we see in the clinic, any amount of tumor shrinkage is actually very helpful. And the majority of patients on the combination arm and indeed the monotherapy arm had some degree of shrinkage, so over 85%. Now, the confirmed RECIST overall response rate, so more than 30% shrinkage, was 10% with the monotherapy, so that's the RAF MEK inhibitor alone. But for the combination arm, in the overall population, so irrespective of KRAS mutation status, it was 45%, 60% response rate in the KRAS mutated group, and 29% in the KRAS wild type patient groups. And also responses to the combination were observed in 3 out of 4 patients which were previously treated with a MEK inhibitor. So I appreciate it's early days, small numbers, but I think this is really encouraging. And in particular, in the KRAS wild type group, where existing therapies that have been investigational or indeed in standard of care haven't shown, to my knowledge, such a response rate.
Yeah, but congratulations on that. So B, I want to turn to Dr. Monk. So, and I do have to call out the elephant in the room. The missing elephant is Dr. Rachel Grisham, who is the USPI for the study and led Milo with you, Dr. Monk, where you did some similar things, trying to break out in an exploratory way this KRAS mutated group of tumors versus KRAS wild type. So can you take us through, I know it's phase 2 to phase 3 comparisons, which is illegal, but just in terms of placing these results into context of what you expect with MEK alone, with KRAS, how do you look at kind of KRAS and the potential of avumetinib as compared to binimetinib?
Yeah, good point. So Milo was a randomized phase 3 trial of another MEK inhibitor, binimetinib, versus standard of care, single agent chemotherapy, weekly paclitaxel, liposomal doxorubicin, or topotecan. A less heavily pretreated group of patients, median number of lines, 2, with a blinded independent central review response rate less than what we're seeing. And congratulations, Susie. Binimetinib has 16% response rate, standard of care chemotherapy 13%. So sort of confirming our historical experience that chemotherapy is ineffective or relatively ineffective. But binimetinib was effective, but not more effective. And so that did not lead to the FDA approval, but it was published in the Journal of Clinical Oncology in 2020. So, Susie, what you're doing, I mean, you just don't do a clinical trial to see what works. You have to test questions. And think of this one, Katie, you're a very, very seasoned and strategic drug developer. So she says, look, we're going to do a trial. We're going to ask two questions. Do you need a combination? Do you need defactinib and avutametinib? And the answer is yes. Two are better than one. The second is that the KRAS mutation is important, a little bit predictive, meaning it predicts the activity of the MEK inhibitor, but very prognostic, meaning these patients do better not only with MEK, but chemotherapy. So does it really matter? And the answer is no. So now all of a sudden we've defined combination yes, biomarker restricted no. And now what we're doing in RAMP is we're trying to sort out the dose, which is a little complicated because the combination is very well tolerated. But in the dose optimus sort of FDA guidance, we're working on that. And then the exciting thing is, I think, and I hope that we'll file for accelerated approval, bring this to the clinic because I have a number of patients like both of you that are benefiting and likely need to do a randomized phase 3 confirmatory trial, which we've done twice before. So this is, quite frankly, this is how you do drug development. You test the molecular signature, the biomarker, you sort out the relative contribution, you dose optimize, and then you confirm in a phase 3 trial. So congratulations, Susie, to your wonderful ASCO presentation. I couldn't be happier with how this program is going.
Yeah, totally agree. I want to talk about, Sue, thank you, Dr. Monk, for talking about one of the two big studies. There are two of them, one with trametinib, one with binimetinib, and they are NCCN listed. So at least in the US, we have them available to our patients with the efficacy that's been presented. I think some of the key confusion points have been the role of KRAS, which we just talked about with this program, is probably, it's kind of like HRD in part, right? It's predictive of like magnitude of benefit, but it doesn't tell you yes, no. And so this is going to be an all-comer development. Before we move to safety though, Dr. Banerjee, can you just briefly tell our listeners, so we've talked a lot about the combination being ideal. The monotherapy is not enough. The combination is ideal. What does defactinib, a FAK inhibitor, bring to avutametinib? What's the rationale for that combination?
So we're going to go back to the sort of molecular rationale, really. So, and some of this obviously is based on preclinical work and also the hypothesis of why MEK inhibitors may not work in all patients and also why they stop working at some point, so the resistant mechanisms. So avutametinib is, as we discussed, a first-in-class RAF-MEK inhibitor. So it inhibits the MEK kinase activity, but it also blocks this compensatory reactivation of MEK by upstream RAF. So the FAK inhibitor, so the issue is that MEK inhibition can also induce compensatory activation of phosphofAK, and that can then stimulate ERK signaling and also can stimulate other growth factors such as YAP and others. So this feedback reactivation, in theory, could be targeted by this dual inhibition of both the MEK and FAK pathways. And there's preclinical data to support that. And I think we've shown here that the combination is better than monotherapy. A question we didn't address in FRAME. We looked at the combination to look at doses there. So I think this is really important. And what's really interesting, I know you're going to come on to the safety, but we didn't see a significantly worse or troublesome safety profile or tolerability with the combination. And that's, again, the importance, as you know, of selecting the right doses going forward when you're combining drugs together.
Absolutely.
The other thing that you did, in addition to the intermittent dosing, but you have a mitigation strategy for the skin adverse reactions. So again, really utilizing what we've learned in our 2 prior trials to make it easier for patients. Sorry to interrupt. Yep.
No, I'm glad you said that because we're going to go to safety right now. And so you just talked about kind of mitigation strategies for rash, which we all know with MEC alone is really, other than the cardiotoxicity, which we worry about, the MEC's what bothers our patients, the rash. They do not like having acne again. And sometimes it can be very, like, not just funny, haha, acne, but like on the scalp, terrible. And so on your study, Brad, let's start with you, Dr. Monk. What was the discontinuation rate from MEC due to toxicity?
Yeah, thank you for that. And it was significant. It was 31%. So, we need, not only more active, but we need, better tolerated medications. So that's
so do we have that? Do we have that with the vudametinib and defactinib, Dr. Banerjee?
So far, which is very much in keeping with the frame. So if we take together the total number of patients, I think it'll be really consistent. So what we saw in RAMP 201 is that around 12% of patients discontinued due to a treatment emergent adverse event. So small numbers and not a randomized phase 3 study, but significantly less than what we've seen with standard of care within both Milo and NRG-281 log study, and also with the MEK inhibitors alone. So in terms of dose reductions, it was around 17%. And thinking about grade 3 toxicities, again, this was numerically and percentage-wise less than what we've seen in the other 2 phase 3 randomized studies. And rash, for example, was under 5% in terms of grade 3 rash or acneiform rash. And again, as you pointed out, the mitigation strategies have really helped, and also us being more familiar with drugs that can cause rash. And so we institute early or prophylactically to minimize this. The main reason, the most common grade 3 toxicity was actually CPK increase, which was a lab abnormality. And that was around 18% grade 3. But now there's been measures in the protocol to actually have dose reductions or interruptions rather than discontinuations due to a grade 3 CPK increase, as patients weren't unwell with this, for example, no significant myalgia.
And have you had, so the other thing we watch for, of course, with the MEK inhibitors, we do echoes and we watch for cardiac toxicity. And I know and I do acknowledge this is comparing phase 3, lots of patients to a smaller study, but any signals that we're seeing that same sort of cardiotoxicity potential with this combination so far?
We haven't seen that so far, certainly not in terms of number of AEs related to this. And certainly in my experience, having done the phase 1 as well, we haven't seen that as what we've seen, for example, with trametinib. But again, low numbers.
Yeah, small numbers. Exactly. Well, wonderful. So it sounds like, just in summary, we have a potentially new combination coming forward. Sort of development plans are still underway. I don't know that we can comment on those just yet, but really actively moving this combination into kind of regulatory enabling trials so we can bring this combination to more women globally struggling with low-grade serous ovarian cancer. And I think I can't emphasize enough the point that you made, Dr. Banerjee, and I think Dr. Monk made it as well. We've had things that provide disease stability, endocrine therapies largely, other things where you have these tumors that just don't shrink, but they don't grow for long periods of time, which is a benefit, and we'll still need them. But I think all of our listeners can probably relate to the fact that a lot of these patients never get surgery because they never have enough response to their chemotherapy, and they just live with the bulk of tumor that they have for as long as they live, as it gets more and more calcified. So the opportunity to shrink tumors appreciably may really help us do and rethink treatment strategies for women, where for low grade, and I know we're running out of time, but maybe you guys want to comment on this. For low grade, the surgical strategy, I'm just going to say something controversial, may be different than high grade. You know, this idea of no gross residual or you don't go, maybe we rethink that. for low grade, honestly. And this drug may actually help us get to a point where we could operate if we were using it earlier on. So I'm really excited about the potential of this. Like, I'm already skipping ahead, and moving it up the lines of therapy. But I think shrinking tumors, we've never shown we can do that in low grade. So that's, to me, a critically important point. I'm going to go to Dr. Monk first, and then I'm going to let you have the last word, Dr. Banerjee, just sort of summarizing the conversation. Any final points you wanted to make, Dr. Monk?
Yeah, thank you. So low-grade serous ovarian cancer is a unique subset of ovarian cancer, very unique, not only from its histologic appearance, but the biology is different. And we can then utilize what we understand about the biology to interact with the genes that are driving it through a clinical trial and see wonderful results, which is showing, at least in the KRAS, a 60% response rate, and even in the non-biomarker, 30%. And these are durable responses, and it's tolerable. So this trial remains open. This program continues to enroll. So please refer your patients if you don't have it open to this trial. And congratulations, Susanna, and great to be with you today, Katie.
Thank you. Dr. Banerjee, last thoughts from one of our international experts on low grade.
Well, thank you, Dr. Moore. I agree with your comments about always thinking about surgery in the recurrent disease setting as well for low-grade serous ovarian cancer. With this particular trial as well, I wanted to point out that the time to response was actually 5.5 months. So don't give up too early. It's important to highlight the responses, and the degree of shrinkage actually develops over time. So I just wanted to make that point. And the final point is, as we've all discussed, this is a rare cancer, and it is such a challenge. I know we've done this as a global community, recruiting women to randomized phase 3 studies. But in a rare cancer, that does take time. And I very much hope we can all work together so that we can get drugs approved in a novel ways, not necessarily having, again, randomized phase 3 trials with treatments that we know aren't as effective as they need to be.
Wonderful. Well, with that, I think we could probably talk for another hour, but we do have to let our listeners get back to their jobs. So with that, I think Dr. Banerjee, congratulations for your leadership. Dr. Monk, thank you for your continued leadership in this space. I'm Kathleen Moore. It was my pleasure moderating this session, and I hope you all have a great day. Thank you.
Thank you.